Okay, molecular biology. So I just mentioned about the few techniques that are used. I'll just go on to now the latest techniques, and I briefly मैंने आपको कहा था पहले introduction part में कि, and again this is just a summary of कि if you want to understand किसी भी चीज़ को, मैक्रो बायोलॉजिकल चीज़ को, वही ना कि आपको पहले समझना पड़ेगा कि वो बनी कैसे है. And these are simple gene level, transcript level, protein level, which is needed because घूम फिर के यही जो bricks जो हैं DNA से लेके ट्रांसक्रिप्ट से लेके जो bricks जो हैं यही बिल्डिंग बनाती हैं, right? तो उसको समझना ज़रूरी है. Now I showed you few techniques. I talked about few techniques which are used frequently. One of the things, how many of you have heard omics, the term omics? Dr. Singh, question. मैं ये पूछना चाह रहा था क्योंकि there is a lot of curiosity among students regarding जो भी उनका कर रहे हैं उससे आगे बेसिस पे वो क्या कर सकते हैं. उन्होंने अगर PhD कर रहे हैं तो PhD के बाद में आगे वो क्या कर सकते हैं, और PhD में जो नहीं है तो PhD उनको कैसे सेलेक्ट करनी चाहिए. So I am happy, ये तो मैं भी करके उसके बाद में I'm happy I'm sit here and ask any question you want, because उसका कारण मैं इसलिए कह रहा हूँ जनरल, क्योंकि many of your questions are common. तो मैं एक एक को बताऊँगा उसके बजाय I can just give you a session and you can ask me any question that you want about your studies and your career and whatever you want to, और जितना मुझे थोड़ी बहुत समझ है मैं आपको शेयर कर दूँगा. But point being कि same question asked by 5 people, I will be giving the same answer 5 times. So I rather give you question once and everybody can then get that. So I am perfectly fine with that. अगर आपकी इजाज़त है तो. So ऐसा करते हैं, proteomics जो है will take another 25-30 minutes or so. जितना भी टाइम लगता है, just I don't want to rush anything. आप नोट करेंगे, I don't want to rush anything. थोड़ा हो मगर समझ में आए, it's more advantageous कि मैं पूरा रश करके ख़त्म कर दूँ, बोले चला गया ऊपर से सब कुछ. So and at any time, as I was saying yesterday, if you don't think जो मैंने कहा है समझ में आया, just raise your hand, please repeat. I missed it. It's perfectly fine. This is interactive. There's nothing for. When I'm standing here, there is nothing formal. You ask anything. It is ये डॉक्टर सिंह जो एफर्ट कर रहे हैं और मैं वहाँ से आऊँ, ये आपके लिए है, हमारे लिए नहीं है. So make sure you take advantage of जितना भी हमें थोड़ा पता है हम आपको बता सकें. Okay, so what I'm going to do is I'll take few slides. I'll walk over, then turn off the camera, and then we'll have Q&A part.
About that one, no issue.
हाँ, उसके बाद में हम इसको बंद कर देंगे, मतलब ये तो आप रिकॉर्ड करते जाइए, फिर उसके बाद में मतलब Q&A, so that people have very good curiosities, which I like. उनके बड़े क्वेश्चंस है, अच्छे क्वेश्चंस है. So मेरे जो आंसर मैं देता हूँ सबका बेनिफिट, they all can get the answer. Right. All right, thank you. So we were talking about this machinery. We were talking about understanding this. So how many of you have heard the term omics? आपने प्रोटियोमिक्स तो सुना होगा, जीनोमिक्स तो सुना होगा, right? So it is basically a Greek word. इसका ओरिजिन कहीं ना कहीं तो है ही ना, which basically mean, as it says here, derived from whole, all, complete. So when you add that om to anything at the gene level, at the protein level, at the transcript level, at the metabolite level, it becomes the whole transcript, whole genomics, whole study of the or the protein level or the RNA level transcription जो होता है. So these are basically the things which are very common these days, which are commonly used from the and then the effect of the not origin, the things that goes through changes, what is known as epigenetic, because of environmental factors. For example, I mentioned earlier, tobacco, smoking, radiation, chemical effect, ये जो भी चीज़ें हैं उसके कारण जो असर होता है एपिजेनेटिक, एपिजीनोम जो चेंज होता है बाद में, उस सब को एट द जीनोम लेवल, स्टडिंग एट द ट्रांसक्रिप्ट लेवल, जो ट्रांसक्रिप्शन होता है, जो चेंज होता है. एंड देयर वाज अ क्वेश्चन ड्यूरिंग द ब्रेक जस्ट नाउ कि कैसे होता है, राइट, फ्रॉम जीन हाउ साइकिल्स यू, आई श्योर यू दैट जो स्टार्टिंग एस फेज, G1 फेज, एम फेज, G2 फेज, ये बेसिकली फेजेस है ना कि आपका जो डीएनए है, जो जो आपकी इंफॉर्मेशन है, इट इज़ बीइंग ट्रांसफर्ड, राइट. हमने खाली उसके नाम दिए हुए हैं बेस्ड ऑन व्हाट वी नो, बट ये सारों को जो है स्टडी करने के लिए किस तरह से करते हैं. सो दीज आर द टेक्निक्स, डीएनए लेवल, आरएनए लेवल, एट द प्रोटीन लेवल. सो एज यू गो फ्रॉम लेफ्ट टू राइट, you are going from larger size to smaller size, right? From DNA, larger, RNA, smaller, protein, metabolite, and then phenotype. So these are the building blocks. Now what are these things? There are 5 different, as you see here I have techniques. They are broken down जो अभी जो prevalent, common, which are commonly used technologies. These are basically 5. Genomic, which is the analysis of at the DNA sequencing level, DNA level. That's the genome, genomics. How आपका gene किस DNA, आपके कौन सा DNA जो आपका जो genes को बनाते हैं, what is the makeup of that? So you understand that, that's your at the gene level. At transcript, at the RNA level, transcription जो होता है, change जो होता है DNA से RNA, जो transcription, जो transcript का makeup क्या है, that is your transcriptomics, analysis of the transcribed RNA, which is, you know, simultaneous measurement of gene expression values in a cell or in a tissue. So with your expression level, and on the left, as you see here, I have in this figure showing कि breakdown कैसे हो रहा है. From here, as you see, it's breaking it down. And then proteomics at the protein level. So DNA, gene, transcription, and then at the proteins, various proteins are from. Then metabolites, identification and quantification of metabolites in a sample. For example, if you have a drug, and yesterday, so I'll tell you how these were useful, because this slide is on the screen. So let me tell you, metabolite, for example, क्या उसका मतलब है, how does it work? जो drug लिया, there are multiple routes. It could be oral, it could be IV, right? Injection through derm. वो drug को लिया, it has to go through, circulate in the body to go to the site of action, जहाँ पे उसका असर होना चाहिए. अब अगर drug लिया, क्योंकि जाता है, and I was telling yesterday, for example, and I showed you earlier today, that if you take the oral route, आप जो फूड लेते हैं सीधा आपके स्टमक को हिट करता है, where pH is 2, break down every single thing. अगर pH 2 पे अगर आपने ड्रग लिया, एज ए केमिस्ट थिंक अबाउट इट कि if you have a molecule which is not stable at pH 2, so what will happen? आपने ड्रग लिया और जो ड्रग की जो एक्टिविटी है उस पूरे स्ट्रक्चर पे है कि ड्रग goes and binds to wherever the site of protein is. If you take something and you require कि पूरा का पूरा molecule as is जो लिया है should go and interact with the protein or you know the biomolecule at the site of action. And if you take something which is oral route, and if it breaks down, metabolite उसके बन गए, and those metabolites are not active, will the drug have its drug function? Will it work? What you need is a whole drug. What you need is a whole molecule. वो तो अब टूट गया. It's broken down to small pieces. उसकी drug function is based on the whole molecule function, because जो भी आपने बनाया है, it is interacting on one side of the protein or the other side of the protein in order to make that protein के ऊपर जो भी protein के functions पे असर करने के लिए. But अगर वो टूट गया, मेटाबोलाइट बन गए, वो बना ही नहीं, तो ड्रग तो काम नहीं करेगा. आपने ले तो लिया बट ड्रग काम नहीं कर रहा. सो मेटाबोलाइट्स को समझना, दैट्स व्हाट इट सेज, मेटाबोलाइट्स इन ए सैंपल, वॉट एवर दैट सैंपल इज ऑफ एनालिसिस, उसके टुकड़े होके, क्योंकि क्या है बॉडी के अंदर एंजाइम्स है ना, देयर आर मेनी different type of enzymes, so when anything that goes in, The enzymes are functioning. They are breaking it out. They are chewing the food, and they are doing whatever उनका functions है. If they break it down to and make something which is not the thing that you want, तो काम नहीं करेगा. So how do we study कि उससे metabolite क्या बने? That is what metabolism is. You study the sample कि उसके broken down pieces क्या हैं. What are the metabolites? Because that helps you understand. And the reason I use that example is because that this is now you will probably will have appreciation of what it takes to bring a drug out. सिर्फ एक डिज़ाइन करके बना दिया means nothing. It is important, but it's nothing. There are 100 other expertise which are needed to, it says it takes a village to raise a child. I don't know if you have heard about the book. कि हर एक को अपना अपना piece contribute करना पड़ता है in order to make the whole thing work, to create the product. So that all of this are very subspecialties within scientific areas. So you have to understand. So that part is known as metabolomics, to understand only the metabolites. And same way then again this micro RNAomics with underlying the control of the transcription. What are the pieces of that RNA वो transcription को जो है control करते हैं. So these now, each one of these have become a full-fledged subspecialty. There are people who do only one of these. There are people who do multiple of this. I think this guy is also doing something. But so that's how. And again, if you look at on the left side, because in order from at the macro level, at the tissue or cell level, to bringing it down, you have to understand all the way to here. क्योंकि जब तक वो नहीं समझ आएगा, सिर्फ आप यहीं पे तक समझते रहेंगे और रूट कॉज ये कैसे चेंज हुए, क्या चेंजेस आए, ड्रग काम नहीं किया, जो मेटाबोलाइट जिस चीज़ के बने वो क्या है, यहीं पे रहे तो उसके रूट तक नहीं पहुँचे, उसकी तह तक जड़ तक नहीं पहुँचे, तो यू डोंट हैव द होल पजल इन फ्रंट ऑफ यू. जब तक पजल्स को समझ नहीं आएगा, जब तक उसकी आपको क्वेश्चन राइट नहीं पूछा है, आपको पता नहीं चलेगा. तो these are the techniques which are used. And now the things that I talked yesterday and the things which I'm going to talk next, in next few lectures, you will see all these are connecting pieces. Okay, how all these things contribute in trying to understand the biology behind. So it's as actually there was a question asked earlier. कि सर आपकी लैब में सारे कैंसर बायोलॉजिस्ट, somebody asked question. Yes, I'm a trained synthetic chemist. I did my bachelor's in chemistry. I did my master's in applied chemistry and did my PhD in catalysis. And I do only अभी मेरी लैब में very small fraction is chemistry. It's 90% biology. All my PhD students are doing are biologists, cancer biologists. And now you'll appreciate that being chemist doesn't matter which field you belong to. Understanding the fundamental, and that's what I piece by piece, not that I know everything, but that's what my curiosity was, to learn step by step, and then slowly things start to come together, they start to make sense. Okay, DNA is just a larger molecule, RNA is just a larger molecule, and not that you understand every single thing, but at least you begin to understand how it might be working. And again, my interest is drug development, so it helps to understand on both sides. And I must say that I have pretty bright students. कई तो यहाँ से गए, इंडिया से गए हुए हैं, and they are among the best. So as I was telling earlier, the difference is whichever thing you pick, you go to उसके जड़ तक जाके ना उसको choose लो, पूरी तरह से समझ लो उसकी. Take few things. You don't have to be jack of all trades. But pick few things and master those, and then slowly start building your building on top of that. Anyway, so these are the techniques which are used to understand how this building is made, what is known as the tissue, the organ, the tissue, the cell. So these are basically few things that I have put here just to show you the appreciation of what it is. And again, मैंने slides इस तरह से इसलिए बनाई हैं क्योंकि you will have all that information. You will have all this information with you. I don't know what their plan is, but all these slides will be shared with them and so you will have. So मैंने कई जगह पे only bulleted items बनाई हैं, कई जगह पे मैंने details बनाई है. So when you read it, you don't have to go to another reference. You can simply just have most of the information in your hand. So that's basically in medicine. So what we are doing is I'm not doing anything beyond that, just trying to show you how various different knowledge bases are used to understand medicine, therapy, develop drugs, or even to understand what human biology or biology in general is. So these are various techniques, these are various tools. है ना कि आपको वो tools भी होने चाहिए ना, वो औजार भी होने चाहिए करने के लिए. Yeah, that's what basically these are. Any questions? Is it simple? That's why there are no questions, or it is so complicated that there are no questions? Okay, I'll continue. Now that I said that you will have these slides. So again, I will not repeat. I prepared this slide just for you guys, so you have simplified version of, what basically genome. ये जो है, it's a systematic study of organisms' entire genome. The human genome, 3 billion base pair. So just imagine 3 billion base pair. उनका permutation combination आप math करके देखिए, right? Or same base pair connected in one way do one type of function, same base pair दूसरी तरह से connect किया है, have a different function inside the human body. See, I was telling you yesterday, बनाने वाले की रचना बड़ी मज़ेदार है. And you have to be just Sherlock Holmes of science. Try to investigate it, and before you know, you just overwhelmed with हर चीज़ कितनी well defined है. So it's fun. The reason I'm telling you is that I'm a chemist, but this is so much fun trying to understand. Again, keep your chemistry hat on all the time. When I'm looking at DNA, I'm not looking at DNA. I'm looking at nitrogen and nitrogen, because that is how I am trained. I'm looking at the base pair amino acid. That's how I am trained. But the flip side, it helps. Because a biologist is looking at the whole DNA को समझने की कोशिश कर रहा है, and I'm looking at a smaller pieces of that. So there are always advantages of what you have, whatever you have learned. So anyway, again, transcript, study of the entire collection of RNA, ribonucleic acid, and what basically happens. So going back to molecular biology. Again, you saw this from the proteomics study. What you need to do is look at on this side. I have just created this, what it does is you could use. So how is ये होता क्या है? Chromatography, you are familiar with chromatography as a chemist, right? Mass spectrometry, you are familiar with that. So basically now there are techniques, very sophisticated mass specs are available, highly sophisticated mass spec. So as a matter of fact, one of the leading lab in the world is Pacific North National Lab, USK, which is very close to where I am in Oregon. This lab is in east of Seattle. That's probably one of the more highly advanced place for omics के लिए. So basically what they do is they are coming up with the technology को advance करते करते कि the resolution से लेके analytical capability is so much advanced कि आप इसीलिए जीन से लेके RNA से लेके प्रोटीन से लेके मेटाबोलाइट उस लेवल तक आप कर सकते हैं. So we collaborate with them and they collaborate around the world. You can collaborate with them basically online उनके साथ में करके. But this is basically it's a mass spec, mass spectrometry जो I'm sure आपके डिपार्टमेंट में यहाँ पे भी होगा instrumentation post में. Mass spectrometry is basically what is used, but it is used to analyze at the DNA level, protein level, DNA level, RNA level, protein level, metabolite level. That's simply the technique. पर अंडरस्टैंडिंग कि आपको जो रिजल्ट्स आया आप उसको एनालाइज कैसे करना, यू हैव टू अंडरस्टैंड बिट ऑफ बायोलॉजी कि क्या हम की चीज़ें करें. पर टेक्निक यूज इज मास स्पेक्ट्रोमेट्री. That's what is used, simple for analysis. So that is, and you can use in combination with the structural analysis, you can use NMR, you can use X-ray which is used. But again, mass spectrometry is the key. जितने भी कहते हैं हम लोग omics करते हैं किसी भी तरह की, basically they are masters of mass spec techniques. But then they study those biological molecules. So they have well understood कि biological molecules at different level, how mass spec, using mass spec, what can be analyzed. So, and this is basically the workflow. You prepare a sample, you separate the protein, select which protein, and you identify those protein. All of this, as you can see here, sample preparation, and the sample could be, again, now you connect this to what I was talking earlier. अगर एक biopsy होती है, एक सैंपल है, पेशेंट सैंपल है, उसकी बायोप्सी हुई, टिश्यू से लिया, जहाँ से भी एक सैंपल को लिया, उसको एक्सट्रैक्ट करके सॉल्वेंट्स वगैरह से लेके एक्सट्रैक्ट करके उसमें से आपने प्रोटीन्स को, सैंपल प्रिपेयर किया गया, प्रोटीन्स को सेपरेट किया गया, एंड देन प्रोटीन ऑफ इंटरेस्ट दैट यू आर, देन यू रन द होल मास स्पेक एंड सी if those proteins are there or not and what is their expression level. And that could be applied. Now, for example, if a protein, you take a sample, कोई भी एक सैंपल लिया, चाहे ब्लड सैंपल है, बायोप्सी सैंपल लिया है, सलाइवा सैंपल है, यूरिन सैंपल, कोई भी सैंपल आपने लिया है, उसके अंदर प्रोटीन्स भरे हुए हैं, आपने प्रोटीन्स को लिया. जैसे मैं आपको बताता हूँ कि हम लोग हाउ डू वी यूज़ इन माई लैब दिस टेक्निक. आपने कोई ड्रग बनाना है या ड्रग बनाया आपने एनिमल स्टडीज़ की. सो यू टेक द ड्रग, यू टेक द सैंपल of a mice or cell line which is पे भी गया है, let's say cancer cell type, whatever the type is, triple negative breast cancer, for example, you take that sample, excuse me, you analyze that and see what proteins are overexpressed. Now you treat with your drug the same cell lines go. Presumably, if the drug is effective, they did something to that protein. Again, you run the mass spec and see what happened, what protein levels change. So you can identify okay, pre and post treatment what happened, right? In order to understand at the gene level or the protein level, at the transcript level, that's what basically you do. So before treatment of the drug, what your sample was, patients, in this case patient sample, this is a very routine. I don't know, probably यहाँ पे PGI वगैरह जो भी medical school वहाँ भी होता है, you take patient sample, you analyze sample, sequencing, NGS, this is next generation sequencing. What is that is done? This is what sequencing, this is what is done. आप उसको analyze कर रहे हैं, तो आपने ट्रीटमेंट से पहले उसको एनालाइज किया तो पता उसमें कौन कौन से हैं मार्कर्स, कौन कौन सी प्रोटीन्स उसमें ओवर एक्सप्रेस्ड हैं. अब आपने उसको ट्रीट किया ड्रग के साथ में, फिर से एनालाइज किया कि कौन से प्रोटीन सप्रेस हुए, और अगर उसका असर होता है देन यू कैन मेक ए कनेक्शन कि इस ड्रग से वो ट्रीट करने से ये प्रोटीन सप्रेस हुए विच वर द कॉज ऑफ द डिजीज. सो नाउ you have some advantage कि आपने आइडेंटिफाई किया. So that's how this technique is used. Same way it could be at the gene level. You can again microarray analysis. Now the techniques are so advanced, it could be on a chip. There are multiple ways to take. एक chip के ऊपर you can have many. You think for example एक जो आपके नाखून है 1 centimeter by 1 centimeter उसके अंदर you can have 1000 grooves. This microarray जो है microfluidics engineering is very advanced. So you take जो chip analysis, lab on a chip and all, जो आपने सुना होगा lab on a chip and analysis on a chip. Basically that's what because each well can have a different type of sample. You can run the sequence. So let's say if you have 1000 genes on a chip, and you take a drug, treat with a drug, and you can see कौन सा gene ऊपर नीचे हुआ with that drug. So you know that drug is affecting that gene or not. Same way it goes with the protein level, transcript level, or whichever. So that's how these techniques, basically that's how these techniques are. And again, as you see here, basically the rest is simply the workflow. But steps are only, So again, I have prepared this slide same way to explain to you what protein metabolite, which I already gave you that. So this is basically, in terms of the knowledge, as you see, This is what I was saying just now. If you did the biopsy, and then you extract and you identify उस सैंपल पेशेंट सैंपल के अंदर कौन सा जीन्स और प्रोटीन्स हैं, बट मास स्पेक एंड प्रोटियोमिक्स बेसिकली हेल्प यू टू आइडेंटिफाई, क्योंकि जब आइडेंटिफाई करेंगे तभी तो ट्रीटमेंट उसकी होगी ना. कि इफ़ यू हैव अ ड्रग एज अ बैक टू द एग्जांपल ऑफ फॉर एग्जांपल 16 डिफरेंट एज अ कॉज ऑफ लंग कैंसर, किस पेशेंट के अंदर कौन सा प्रोटीन जो है कॉज है आपको पता चलेगा, तभी तो उससे मैचिंग आप ड्रग देंगे. अगर प्रोटीन ए के लिए ड्रग 1 काम करता है तो प्रोटीन बी से लेके बाकी 15 प्रोटीन उसको ड्रग 1 देंगे काम नहीं करेगा, तो पेशेंट बेचारे को आप स्ट्रॉन्ग क्या देते जा रहे हैं, उसका असर हो नहीं रहा. सो दैट्स व्हाट बेसिकली पर्सनलाइज्ड मेडिसिन. यू आइडेंटिफाई कि व्हाट इज द कॉज, विच प्रोटीन इज द कॉज ऑफ द डिजीज, and then you see the matching drug in your kitty, in your toolbox that the physician has, then you try to give. But the techniques, these are the techniques which are used. So if you look at here, right में जो मैंने जो cartoon जो उसका है, जो regulatory mechanism for example, basically what is happening is, you know, from the epigenetic causes, splicing करके protein, post translational protein level पे, gene level पे, transcript level पे, protein level पे. So you keep on analyzing these. But जो technique use, जो omics जो use होती है, this is what is done. This is a biopsy, same thing could be blood sample, saliva sample, urine sample. But these are the techniques. Basically, you run, you identify. So आप ये डब्बा आपने देखे होंगे, ये डब्बे, right? आपने instruments देखे होंगे. But behind the scene, that's what they are doing. वो तो ठीक है ना, it's a pretty box created. But inside, that's all it is happening. They are just breaking it down to protein and just simply जो भी signal है, signal is captured and that what you see, and then based on that you can identify which protein is present or not. So this is basically, you know, the technique, sampling and techniques which are used. The flow chart is the same. I just wanted to re-emphasize. If you look at this, क्या है sample? It could be any sample as I mentioned. You create gel as I was telling you earlier. Use proteomics, again basically mass specs is, and identify the protein. Now this is for the quantitative proteomics. As you see, it helps to understand kitni cheezein, what is the complex mixture of your structure. It helps to understand the spatial arrangement in the microenvironment inside the tissue or inside the cell. It helps you to understand drug mode of action, MOA, mode of action, mechanism of action of a drug. Targets, जो example I told you just now, you would take it before and after treatment. If you look at that, that tells you the drug is working on which genes or not. So it tells you the, you know, drug action. It helps you, in understanding of what signaling pathways, how things are degraded inside the body or the cell, and what disease biomarkers. What are the causes of the disease, the biomarkers, the proteins, the genes जो mark कर सके, identify कर सके. Epigenetic, what changes happen? I gave you example of tobacco, for example, radiation. Radiation attack, chemical weapons, quote unquote जो हम use जो सुनते हैं, अगर वो chemical weapon, because they are used as a weapon, but that chemical that went and affected, interacted with the biological system, what changes happen? You can learn that again using proteomics and complex system inside the human body. You can still see how important and useful tool it is, because it is now the central to any biological, human biology understanding, drug discovery, translational research, and again the topic of our course is experimental therapeutics. So in order to understand what therapy to create, how therapy works, you need this tool, and this is one of the most advanced tools. So see, it helps basically, you know, experimental therapeutics. This is now, this is just a summary of what I have been saying for past some time. This is how this omics, now see how it is connected. आपको पता चले बायोमार्कर क्या है, उसको आइडेंटिफाई करने के लिए could be a blood sample, tissue sample, urine, saliva, जो भी body fluid है, you can use inside, you know, any human part. This is just showing brain का, for example, right? क्या changes आते हैं. You can subtype identify population, जो मैं कह रहा हूँ, data science, big data, all of that information, all sequencing done upon, let's, for example, again, I use the same example again. If there are 1 million people जिनका